Okay. Yes. Welcome again to my channel. My name is John Bosco, and I'll be taking you through growth media used in microbiology for culturing bacteria. So, first to study the bacteria and other microorganisms, we have to know how we can cultivate them and grow them. So it is necessary to grow them in controlled conditions in the laboratory. And growth media is an environment that contains all the required nutrients for bacterial growth. So normally there are two types of growth media. That is, we have the liquid media and solid media, whereby liquid media is called a broth, while solid media is called agar. We can also have a semi-solid media, and you can see a picture here. Yes, you can see a picture here. We have a solid agar slant. Then we also have broth media in the test tubes. So we have to know that there are other forms of presentations of agar, where we can also put them in we have agar slant, we have a deep slant, and we also have agar plate. Now the agar plate is this one. Yeah, this one with pinkish color. That is the agar slant. So let's study these presentations, solid growth media forms. We have agar slant, and we have agar deep, and also we have agar plate. Now, what is the advantages of using solid media over liquid media? So in solid media, the bacteria are able to exhibit certain characteristics that is required, therefore making it easier to identify a bacteria. So we can see here we also agar slant. It is usually used for stub culture, and we can also see there are two types here. There are two examples, two diagrams drawn to represent agar slant. Now, broths can be used to determine growth patterns in a liquid media and for certain types of inoculations and metabolic tests. They can also be used they can also be used as a method of choice for growing large quantities of bacteria. Now, in agar slant, they are commonly used to generate stocks of bacteria, and agar plates can be used to separate mixtures of bacteria and to observe colony characteristics of different species of bacteria. Therefore, it is usually advisable when you want to identify certain bacteria from another. It is usually advisable to use agar plate. Now, dips are used for several different types of dips are used for several different types of differential metabolism tests, e.g. the caseins hydrolysis test. So, types of growth bacteria, having learned that there are either two or they can be three, but normally we have two major forms, where we have a solid media, and we have a liquid media, and also we can also have a semi-solid media. Now let's look at the types of growth media. Yes, so growth media can be categorized based on their chemical constituents or the purpose for which they are used. So if we can also let's begin with one. We have complex growth media or rich growth media. Contains ingredients whose exact chemical composition is known, e.g. blood yeast extract. We know the components of this growth media. The ingredients, we know exactly the amount and the type of ingredients that are found in this media. So for example, if we look at lactose broth, we know the what is, for instance, we can have peptone, what is found inside this growth media. For example, in yeast is usually found in LB media, lactose broth. Now the second one is synthetic or chemically defined growth media. These are formulated to an exactly defined chemical position, e.g. a general purpose growth media like TSA, that is tryptic soy agar, or Loria broth, LB, is used to grow a wide variety of nanofastidious bacteria. This type of media is often a complex growth media, and you can see in these images how these things they look like. So we can also move to specialized media types. Selective growth media contains chemicals that allow some types of bacteria to grow while inhibiting the growth of other types. An examples of a purely selective media is the PEA, that is phenyl ethyl alcohol agar, which allows gram-positive bacteria to grow while inhibiting the growth of gram-negative bacteria. We can also have McConkey agar. McConkey agar allows only the gram-negative bacteria to grow, but inhibiting the gram-positive. We can also have, now if we look at this image, we can see Staphylococcus aureus, a gram-positive organisms, grows in this PEA plate, while Serratia marcinsens, a gram-negative, does not grow. Alright, so let's move on. A differential growth media is formulated such that different types of media will grow while with different characteristics. And we have to know here, we usually consider the color of the colonies. An example of a different growth media is blood agar, which differentiates among bacteria based on their ability to break down red blood cells and hemoglobin. Blood agar is also a complex growth media because it contains blood. You can see that one is that is a picture of blood agar prepared. That is a swab. That is bacteria. It has been inoculated. Now, our growth media can also be selective and at the same time differential. And we have an example here, EMB, that is inosine methylene blue agar. This type of agar inhibits the growth of gram-positive bacteria. Gram-negative bacteria will grow on this media, but then will be differentiated based on their ability to ferment the sugars, lactose and sucrose. Strong fermenters of lactose or sucrose will produce large amounts of acid and will appear dark purple to black. E. coli, that is Escherichia E. coli, a strong fermenter will frequently appear as colonies with dark black with a green metallic sheen. Weaker fermenters will produce pink colonies. Now, if you find clear or colorless colonies, this will indicate that the bacteria do not ferment either sugar and is not a fecal coliform. Okay, so you can see that picture. That is image of EMB agar that has been inoculated with E. coli. And E. coli is a gram-negative coliform bacteria. Now it has shown here growth of dark blue-black colonies with metallic sheen, indicating vigorous fermentation of lactose and acid production, which precipitates the green metallic pigment. So we will take you through procedures of making and bacteria. Just let's look at this table here. Mannitol agar. Now, if you see a growth in mannitol agar, what does it mean? So the growth organism has not been inhibited by sodium chloride. If there is no growth, they have been inhibited by sodium chloride. Now, which type of bacteria grows in mannitol agar? Mannitol agar is mannitol salt agar, that is MSA. It is usually differential and at the same time selective. So, Staphylococcus and Micrococcus. If there's no growth, it is the bacteria is not Staphylococcus. Now, because it is both differential and selective, we can also see in differential if there are colonies, but then the colonies are yellow, it means that those organisms ferment mannitol. And then we have no, if there's no yellow colonies, those organisms does not ferment mannitol. So example of those bacteria that ferment mannitol is S. aureus, and those that do not, they grow but do not ferment. There are other Staphylococcus species, but not S. aureus, because S. aureus will ferment mannitol. Micrococcus will show yellow colonies. So, EMB agar, that was mannitol. So let's look at the EMB agar. If the result, there is no growth, it means that it was inhibited by the dyes that are present. And we all know that EMB agar contains peptone, lactose, sucrose, and the dyes, eosin Y and methylene blue. So if there is no growth, it means that those organisms were inhibited by dyes. Now, interpretation, it is gram-positive. Remember, EMB agar only allows growth of gram-negative. Alright, so if there is growth, organism was not inhibited, there is no inhibition, right? So that is definitely it will be gram-negative. Now, if there is growth, but then colorless growth, organism does not ferment sucrose or lactose. Therefore, it is non-coliform. It is non-coliform, yeah. We can also, if the result, if it's pink or mucoid, the organism ferments lactose or sucrose with some acid production. Now that's the coliform bacteria. If there is a colorless colony, there is no, the bacteria is non-coliform. What about if growth is dark or purple to black with or without green metallic sheen? Organism ferments lactose or glucose with large amounts of acid production. That's ID. And then interpretation is E. coli, direct. So thank you for watching this video on this growth media. And if you want more information about other microbiology tests that are done in the lab, don't forget to share and like and comment so that we can move on together. Thank you very much.